Okay, Assalamualaikum and hi. So before we continue with our next subtopic, so let's have a thinking corner. Okay, so in your study, what do you think one factor that makes you study hard? Okay, what is the one factor that makes you study hard? For me, the factor that makes me study hard is because of I want to make my family proud of me. So that's why I study hard. So what's about yours? And how can you relate that with enzyme? Okay, how can you relate this situation with enzyme? So keep your answer and you will write it in the link of task that I will give later. Okay. So we will continue with the enzyme part 2. Huh, in your opinion, how do you think enzyme was formed? Okay, how do you think the enzyme was formed? Okay, keep your answer and you will write it down in the link that will be given to. So first, intracellular enzyme. So what is it? Intracellular, as you can see, the highlighted one in, okay, we can remember it by inside a cell. In for inside a cell, in a cell. So intracellular enzymes are enzymes that are synthesized in a cell for its own use. Okay, it will use for itself. They are used for biochemical reaction that occur within the cell. Okay, we can make a resemblance that we cook for ourselves, we cook for ourselves for us to eat, okay, not for the others. So that's how intracellular enzyme, and they are found in cytoplasm and nucleus, for example. So example of enzyme in cytoplasm, we have hexokinase, glucoisomerase. In nucleus, we have DNA polymerase. In chloroplast, we have ribisco. So extracellular enzyme are enzymes that are secreted outside of the cell. If you can see the highlighted one, X is exit, okay, meaning that it will exit the cell. So it will catalyze the biochemical reaction that occur outside of the cell. Okay, meaning that, for example, I cook, okay, myself, but I give it to the others for the others to eat. Okay, that's how extracellular enzyme. So for example, is trypsin enzyme that is produced by pancreatic cell and secreted into duodenum to break down the polypeptide. And also we have amylase and also pepsin. So now is the production of extracellular enzyme. First, ribosome is the site of the protein synthesis. So the protein synthesized by the ribosome will enter the lumen of the rough endoplasmic reticulum and are transported through it. So if you can see, okay, here is the ribosome. After it synthesizes the protein will enter the lumen of the rough endoplasmic reticulum, or RER, okay, and it will transport through it, okay, along the RER. So after it been transported, once it reach the end of the RER, the membrane buds off to form transport vesicle. Okay, here if you look at the number 3, here it reached the end of the RER, the rough endoplasmic reticulum, and it will bud off and become transport vesicle. Okay, the number 4. And the transport vesicle that contains protein move towards the Golgi apparatus and then fuses with it. So after it become transport vesicle, it will go to the Golgi apparatus, okay, and then fuse with it. It will fuse in the Golgi, okay, apparatus. The protein will be modified into enzyme and secreted in secretory vesicle that are formed at the tip of the Golgi. So after it had become, the protein transferred into the enzyme, and it will from the tip here, okay, it will become the secretory vesicle. So the secretory vesicle will move towards the plasma membrane. Okay, so it will go to the plasma membrane and fuse with it, and you can see that the dot dot one is the enzyme. Okay, it will secreted as extracellular enzyme. So mechanism of enzyme action, we have lock and key hypothesis. Okay, so most enzymes are complex proteins that made up of polypeptide chain that are folded into 3D structures, and the 3D structure has an active site that complement the substrate molecules. Okay, if you can see the diagram here, okay, this is the polypeptide chain that are folded into 3D structure of enzyme. So the purple one is the active site. If you look at the diagram, the right one, okay, this is the 2D diagram of the enzyme. So here the active site, okay, this is the resemblance of the 3D. Okay, so let's think. For why? How do you think lock and key are related to enzyme and also substrate? How do you think lock and key related to enzyme and substrate? Write down the answer also, okay, in the link. So the binding of substrate to the active site is said to be lock and key, where enzyme represented by lock and substrate represented by key. Okay, if you look at the diagram here, okay, this is the key, this is the key, this is the lock. Okay, and enzyme is as the lock and substrate is as the key. So why we said it as lock and key hypothesis? The shape of the active site of enzyme are exactly complementary, or it suit with the shape of the substrate as represented by lock that needs a key that fits, and only then it can be opened. Okay, let's say it's like this. When you want to open a lock, okay, maybe your house lock, so you need to have the key that fits with it. Only then you can open the lock, right? You can open the door, for example. So that's how is it. If you use the wrong key or the key that do not complement with the lock, it will not be open, right? So that is how the enzyme and also the substrate that only fits with the active site, okay, is the one that can bind with the enzyme. So the last one is the energy diagram. So here we have two curve. Okay, so activation energy is the energy that is needed to break the bond in the substrate molecule before a reaction can occur. Okay, meaning that before we want to do one reaction, for example, we need to have this amount of energy to reach or to perform